So without further ado, I'll introduce Jim. For 45 years, Jim Kane has had the good fortune to study diverse native bees, their nesting and foraging ecologies, their propagation, and their pollination of both wildflowers and a variety of crops across diverse biomes of North America, including such Northwest crops as blueberry, cranberry, and alfalfa for seed. He is retired from the USDA ARS Bee Lab in Logan, Utah, but continues field research and publication activities. Across the Great Basin, he has studied pollination and pollinating bees of a dozen prevalent wildflowers, including one or more species of astragalus. Oh man, these are gonna get me. Balsamorrhiza, uh, Connectus,
Yep.
Clomi,
Cleome. Yeah.
Cleome, Lamatium, Penstemon, uh, Phacelia,
Yep.
and oh man, that's so
Spheralcia.
small. Say that last one for
Spheralcia.
me, Jim. Thank you.
Spheralcia.
I appreciate it. It's so small on my phone here. Uh,
Oh,
Other
yeah.
studies include bee chemical ecologies, larval and adult dietary needs, uh, kleptoparasitism, parasitism, excuse me, foraging specializations, and large-scale studies that explain regional and temporal variation in bee abundances and diversity across years, decades, and millennia. He most recently spoke as a plenary speaker at the big biennial native bee meeting held north of Sao Paulo, Brazil. And without further ado, sorry for mispronouncing all of those, Jim.
I
Uh,
can understand on a cell phone screen.
Too small for me. Uh, Go ahead and begin, Jim.
All right. So, um, as you said, I'm Jim Kane. This picture is of lupins flowering north of the Jarbidge Range in northeastern Nevada, area that I've come to appreciate for its beauty, at least in the springtime. It gets fairly dry the rest of the year. All of my work in my career, except for a little bit of work with sweet onions in Walla Walla, all of my work has been with dicots, such as these lupins. And but there's been a couple monocots that have puzzled me, one of which I worked with while I was still working with USDA, and the second one you'll see is one that's puzzled me all along, and I was never able to be around in the spring to work with it. And that'll be fritillaria. We'll see that about halfway through the talk. Death camas all used to be in the genus Zygodinus, very tidy, and goes back, the name comes from the nectaries. You can see them, they're kind of in a green color, and you can see some reflection off of accumulated nectar. This is on a different species, and it's self-explanatory there, I guess. Um, but this has been messed with now. Zygodinus has been broken back into several genera. This one's been moved into Anticlea, still the species elegans, and I won't talk about it tonight, although I've worked with it quite a bit, especially at high elevations where it attracts a remarkable diversity of flies. The one I'll talk about tonight is one we mentioned earlier, which is Death camas. Um, and this is now in the genus Toxicoscordion. Um, and it's been moved out of the Liliaceae, which everyone knew the Liliaceae was quite a catch-all anyway. And it, along with Anticlea, have been moved over to the Melanthiaceae. Is the same genus as Trillium, which it doesn't look like at all superficially, but in fact, some of the floral characters fit well with Trillium, and the molecular evidence backs that up. And it's well named in that essentially it's death garlic, if you go by the Latin name. And indeed, I'll get to that in a little bit. And this is the one, there's a second one, venenosum, that I have my doubts whether it's a separate species or not. The separating characters are quantitative and a lot of overlap. So this is true camas, and you have camas meadows like this out in western, um, maybe all through Washington State, but certainly in western Washington State. And as you know, this was extensively used by native peoples for their starchy bulbs to eat as a energy source. And although the flower looks vastly different from the death camas flower, when you're digging up the bulbs in the fall, they might look quite the same. And death camas, unlike this one, death camas is toxic. It's poisonous. Work at the USC Poisonous Plant Lab here has shown that it's poisonous to cows and sheep. And we know it's not good for people either. You can see these cows on an overgrazed BLM allotment in Nevada. And you notice they've eaten all the grass down the nubbins, but all the stick-ups, I have one of them circled there in red, those are all death camas flower heads late in the bloom cycle, and the cows aren't touching them, which is probably also why they're multiplying in this pasture. And you'll see why in a little bit, why they would know straight away that it's toxic. Now, there are many, many plants who are chemically defended. It's one of the primary ways in which they reduce herbivory. This one, for instance, creosote bush, is a prevalent shrub of the warm deserts of Western North America and Northern Mexico. It's a dominant shrub, actually. It's a defining plant of the Chihuahuan Desert. And its foliage is laden with resins that are so toxic that the only things that feed on the foliage are specialist insects who feed on no other plant. You don't find generalist herbivores feeding on creosote bush. But if you turn to the bees that visit it, and I've worked with them along with several colleagues for almost a decade, there's an enormous diversity of bees. There's a number of specialists, but they're specialists for the same reason that they're specialists on willow, which is that this plant blooms so reliably in such abundance every year that it's all they need to know. And so there's specialists like the ones pictured on the right, but accompanying them are about 100 species of generalist bees, all the way out there, including honeybees, that merely visit these flowers and collect the pollen and nectar without harm. And this is a theme over and over again in flowering plants. And it's why you can't look at a plant and say, oh, that's toxic for deer or that's toxic for me, therefore it must be toxic for bees. It just doesn't work that way generally. And then we have the prominent exception, perhaps now the best known exception available, and it's with death camas, Toxicoscordium paniculatum, at least. People may be able to add some other species to that out where you are in Washington. And the unfortunately named Andrena astragalus. Now, the name astragalus came up in that discussion a little while ago. And when TDA Cockerell first identified this species from a specimen he collected it at astragalus. And this species, thus far, has never been collected at astragalus since. And it's one of the risks of naming a bee after a plant species without knowing enough about its floral visitation behavior. The pioneer work with this, well, it was a couple people. One was Hitchcock here, but also Vince Tepedino, who was the one who first, I believe, associated Andrena astragalus with death camas. He couldn't continue that work because USDA said, well, what's the economic benefit of this? Stop that research. And he was pulled off of it. But I was allowed to pick it up some years later when I came to Logan, as long as I did it on the side. My collecting experience with the plant and the bee is illustrated by the red stars. And because I'm a lot further east than you people, it's centered in northern Utah, northeastern Nevada, out into southeastern Oregon, and a little bit over into Wyoming. I've not collected at death camas in Washington State because I could never be out there early enough. So what you people find will be really valuable to know, and make sure you have a good sense of the species you're looking at, because I don't know all the possibilities there. But at all of these sites, inevitably, the only visiting bee was Andrena astragalus and was present in all of these sites. It's rare in good weather with full bloom that I don't find the bee within 5 or 10 minutes of looking. Importantly, because that tells you that it visits the plant and other bees don't, but in order to know that it is a specialist, it just so happens that the reason I was in all those places is working with this variety of native wildflowers that are wanted by BLM for commercial seed production for reseeding back into basin habitats. And so Collected a lot of bees with this, identified a lot of bees with this. And of the 3,800 individuals across a decade of work, I never saw Andrena astragalus at any of this diversity of flowering plants. So that suggests to me that it's quite the specialist. There may be some things in the Liliaceae, although I haven't seen it either at Allium, the wild garlics. So I suspect it, in fact, is a very narrow specialist, at least so far in the areas that I've surveyed. Now, one of those plants I've surveyed extensively is Astragalus philippes. And as you remember, I said that Cockerell originally identified this bee from a specimen he collected at Astragalus. And this one also has white flowers, and it can be really abundant, upwards of 500 to 1,000 flowering individuals at a site in good sites. And it's unusual amongst the genus Astragalus that a little bit of work, you can identify it in the field. A lot of Astragalus will drive you crazy trying to identify them. It's the largest genus of flowering plants in the world with some 3,500 species, with a huge diversity in Iran of all places. And I've gotten a lot of bees from Astragalus philippes. This is one thing I found exciting about if BLM would ever get around to actually buying seed from growers to plant and actually planting it back out, is it feeds enormous diversity of bees. That's almost a third of the Osmia species, well, a quarter of the Osmia species of North America can be taken at this one species of Astragalus. All the bumblebees of the basins that I've collected in are at this plant one time or another, and a number of User as well as other bees, but never Andrena astragalus. And yet, because that could be the case, and this does occur in flowering plants. There's flowering plants like dandelions that don't need a pollinator. They merely set seed with no visitation at all. That's not the case with toxic ascordion, though. With this death camas, this foothills death camas, it needs a visitor, and in fact, it needs an outcrosser. Our manual outcrossing is equivalent of freely visited flowers, but perform much better than where we self-pollinated, cross within flowers within a raceme, or we didn't allow any visitation. Most of the capsules never formed from those flowers. And when a capsule did form, they were usually depauperate in seeds or were empty. So the plant, a pioneer seed of this plant, arriving in a new spot, may be able to set some seed, but it's going to do a whole lot better once it's got some progeny around it for cross-pollination. And this is the primary toxin, zigacine. And it works by blocking open the synapses of neurons. That is the little gap between nerves across which chemical neurotransmitters travel to continue a nervous impulse. And that is something that we depend on, not only firing, but then shutting down. And what zigacine does is it blocks that voltage gate open, such that the nerves just constantly fire without end. They never come back to a resting state. I mentioned that there is a case of at least one that was in a newspaper account from down in St. George from a couple decades ago of a woman whose friend brought her some wild garlic bulbs to eat. And it turns out amongst them were some death camas bulbs. She bit into one, and she lived to be able to tell that she felt a tingling sensation on her tongue before she chose to swallow the bulb. Her husband came home, found her on the floor, trembling, got her to the hospital. They pumped her stomach. She was okay. But this is the only, I've been tempted, but I'm not going to do it, to see if feel the sensation for myself. But it suggests that it's messing even with peripheral sensory nerves. And this will be important in the story that I will present. And that's because bees that sample the flowers likely are, well, you'll see. So I've never seen a nest of Andrena astragalus, nor has one ever been reported. But I needed to evaluate that, well, is this really toxic? Because remember the example of the creosote bush in which the plant is toxic, but the nectar and pollen are not. And so since I reared quite a few Osmia lignaria at home, I had lots of surplus males who I could freshly emerge, starve for 24 hours so they were good and hungry, and then put them on freshly cut racemes of Toxicoscordion paniculatus and watch what they did. And you can see this male, he knows he's on a flower, he's hungry, his tongue is out, and he will proceed to drink from a couple of flowers, drinking the nectar. And just to make sure that I hadn't messed with the bees and weakened them by starving them for 24 hours, I also presented them with mint or apple flowers to watch how many flowers they visit. Can they continue to drink? Do they fly away? Are they happy? This is my control. What we found is that fairly soon after probing flowers, so after probing a couple of flowers, most bees would stop drinking, and I'll show you a video at the end that shows them manically wiping their extended tongues. They don't drink anymore. The few cases we've had is after they've had this one experience, they refuse to drink anymore from these flowers. They've learned their lesson. A few individuals who continue to drink, within a minute or so of drinking the nectar and getting a bigger dose, trembling, they fell to the cage floor, trembled there for a while, and died. So this is a toxic plant for at least this generalist bee, and as far as its nectar goes. I was curious then about the larvae. Maybe this was a response of just the adult bee. And so with all the females I had flying at the house, I designed and implemented this observation nest that I could get into easily in order to dose fresh provision masses with eggs with known concentrations of death camas that had been isolated by a chemist here for me for assay. And so I had a number of nests and we could dose them all with a known concentration and then look at larval response. By using food dye, we showed that the liquid would spread to about a third of the provision mass. So not every larva would encounter it at the beginning, but as it fed through the provision mass, ultimately it was unavoidable. And this is just a feeding larva in one of those nest cells. What we found is that at the least dosages, the bees survived just fine. And what this graph is showing is the number of bees that finished their provision mass and proceeded to spin a cocoon. And what you can see is that the higher doses there, and those are in microliters, so it's very little, or micrograms, it's very little. They never finished feeding and they died before spinning a cocoon. And what I could see under a scope with those observation nests is that the larva, days before it died, it would rock back from the provision mass and just sit there. It ceased feeding, whereas otherwise the larvae are constantly feeding, but they refused to feed. I imagine they were sickened before they died, and that it foreshadowed the fact that they would die a couple of days later. So this pollen and nectar of this plant is lethal in a provision mass. And from our calculations, it's that about if a quarter of a provision mass of pollen and nectar includes pollen and nectar, a quarter of it is pollen and nectar from death camas, it will kill a lethal dose for larvae. So this is not a desirable plant to be feeding to your offspring either, even if you avoid drinking nectar from it as yourself as an adult bee. Now, the question of nomata came up, and I imagine a lot of you folks now recognize nomata being such a gorgeous little bee. This is not from Andrena straguli, because like I said, I don't have a nesting site. This is from another Andrena that nests here, a large aggregation, and they were foraging on wild cherry flowers. And I could bring a bunch of them home, starve them for 24 hours, and then let them feed on death camas flowers. And they were even more susceptible than were the osmia. Just a few flowers, and then they would tremble, shiver, drop off the flower, unable to hold on to it, and go into this contorted death pose and die. And this may be a benefit of Andrena straguli being adapted to tolerate or eliminate this toxin, in that it can make provision masses that will kill nomata. And so if I ever find a nesting aggregation, you can bet I will be looking to see, are there any nomata parasitizing this aggregation at all? Because it may be that this is the way in which Andrena astragali sheds one of its nemesis. Don't know that. If any of you find a nesting aggregation of the bee, mark it, take a GPS, and be prepared to do some study. It would be fascinating to know. So that's just a summary. Um, and it's pretty much everything I've told you to this point. So it's an unusual plant. At the moment, I think it's among the best demonstrated examples of a plant that has a monolectic bee who tolerates a toxin that kills all other bees, both as larvae and as adults. Now, the topic of syrphids came up, also one of your comments. Um, and syrphids, it's true. I confess to a bias for bees, and I've had that bias for a long time, partly because it's only been in the last 5 years that there's good online keys to the genera of Syrphidae and to the species of Aristolus. But those keys are nice. I've really been enjoying them. And it turns out there can be these flies in abundance. One of the nearby canyons here that faces out into the suburb, this one Aristolus arbustorum, I found several hundred of them working the flowers. You can see that one's tongue is out, probing for nectar. When I dissected several of them, there was pollen in the gut that was resembled that of Death camas. They were all male syrphids. When you were talking about large eyes, in the case of this species, the eyes meet on the male, the eyes meet above the top of the head. They were all males, and it's a European species. So it's not adapted for Death camas either, because the genus Toxicoscordion is limited to North America. It does not occur in Europe or Asia, which is where this fly's predecessors came from, probably in the last century or two. So that's a big puzzle. This would be worth following up on for sure. And where you do come across flies visiting the flowers, collect them. And you also may be able to identify them. I can easily provide that reference if you can't find it yourself. So this is another view of a different place. It's on the Wine Cup Ranch in northeastern Nevada. Gorgeous, gorgeous location. The ranch is about 300,000 acres. It got its name from when Jimmy Stewart and two other Hollywood stars bought it as a retreat back in the 1950s. It's still in private hands, but the flora in a good deal of this ranch is in spectacular condition. And growing in those contexts and flowering in early spring, as does Death camas, are two species of a true lily. This is genus Fritillaria. And Fritillaria, we locally have two species. On the left is Pudica, which it does attract bees, Habropoda, perhaps Anthophora, possibly others. I've not studied it. And on the right is one that's puzzled me for years. I'd only come across it several times, but you have to admit that's a bizarre flower. Maroon and cream-colored speckling. And that's Fritillaria atropurpurea, also known as a checker lily, sometimes called a chocolate lily, though it's not any chocolate that I would eat. And it has some similarities with Death camas. It comes up from a bulb, it's a perennial, it forms a capsule, it's a monocot. But some of the similarities disappear after that. For one, Fritillaria occurs all around, the genus occurs all around the Northern Hemisphere, and it has been a hotbed of research in recent times, especially in China. And then this website, which I would strongly recommend you visit, even if you're not that interested in plants, it's downright artistic. It's beautifully done by Lawrence Hill. He calls it Fritillaria iconis, and it has pages of plates of flowers, of bulbs, of phylogenies, of individual species accounts. And I don't know whether he's independently wealthy or what, because he's visited a lot of places in remote spots like Tajikistan in order to find representatives of this genus. The biggest one, and you can buy it as an ornamental, is that huge bulb that you just saw is Fritillaria imperialis. And I was sent these pictures by a colleague in Iran. I can't, it would be fantastic to come across this plant in the wilds here, but that's not to be. And needless to say, a US citizen is never going to be in Iran to see this, unfortunately. But nonetheless, there's still a lot of other spectacular species, as you can see from this slide, a great variety of floral morphologies among the species, including repeated over and over again, this kind of a color that's the maroon with the cream speckling. It shows up in Europe. It shows up in China, Japan, Turkey, and Iran, at least. Maybe other places too. Russia, for that matter. I didn't list that there. The most common one and the ones in cultivation as well is Fritillaria meleagris. I have some just coming up at the house now that I planted last fall from commercial stock that's propagated. You would think with that kind of a flower color and morphology, especially the color, you might expect carrion flies. And indeed, that's a carrion fly there on that flower that was sent pictures from a colleague in Japan. So I finally been able to work with it. And I have a population fairly near our house at the mouth of Green Canyon. And this population has been there since at least the 1950s. I was led to it by a student who's about my age now, who was an undergraduate when he took general botany, was led to this hillside to show off this plant on a field trip. It's a really steep hillside. It's about a 35-degree slope. So it's one of those kinds, if you dislodge a cobble of rock, it starts bounding downhill with bigger and bigger bounces the further it goes and eventually reaches the bottom, bounding about 6 feet into the air. So it's not a place I want to go tumbling. But overall, I was able to find about 70 plants flowering on this overall hillside. I've marked 5 of them there with arrows, and those have pollination bags on them for the manipulations I was going to do with them in order to first understand what the breeding biology of this plant is. Does it need a pollinator even? Does it need outcrossing? My initial perplexity, however, was the first flowers I encountered looked like this. You can see the stamens with their pollen-laden anthers, but where's the pistil? I couldn't find a pistil to save me. I was wondering, gosh, are there specialist herbivores that just eat the pistil? That seems ridiculous. Am I losing my touches, botanically speaking, and can't even find a pistil in a flower? Well, as I looked around more and found more plants, I did find the pistils. And you can see they have these 3 big reflect styles with a stigma on the end. And the stigmas are in the same plane as the anthers. So an insect tall enough walking around inside of that flower hits both. So it's a bisexual flower on the left and a male flower on the right. Now that could be that the species is in fact andromonoecious. Maybe there's male individuals and bisexual individuals in the population. So I looked around some more, and with some effort, I could find plants with multiple flowers, including ones like this that has a male flower and a bisexual flower. So the bulbs themselves are bisexual, or at least most of them are. And from measuring the size of the plants, and this has been shown by an Italian group that you can estimate it by the diameter of the flowering stem, I was able to show that the smallest plants start out with 1 male flower. A little bit bigger plants have 1 bisexual flower. Bigger plants have multiple flowers, up to about 5 or 6 flowers. So it's in fact that all the plants seem to be hermaphroditic, although I'd have to actually track one for over years to make sure of that. And it's something called gender diphasy, in which they start in a male phase and eventually enter into a bisexual phase. And a little bit more dissection I could find in the male staminate flowers down between the filament bases of the stamens. There's a little bitty pistil down in there, a little dwarfed pistil that's not going anywhere. And so the evidence is there that even the staminate flowers are in fact have this, they're actually bisexual, they just aren't functional as so. Again, from Lawrence Hill's website, here's a selection of different pistils of different species of Fritillaria, and you see that ours is one in which that style is divided for most of its length, unlike some of these others. So back to my pollination experiments with the bagged flowers. I pollinated them, did outcrossing, selfing, no visitation, open visitation, and pollen supplementation. And then later on collected the capsules, not when they were ripe like this, because I was afraid of losing the seeds, but when they were still green but fully sized. And this is where the name of the plant comes from. The fritillaria, or fritillary, is the leather sewn dice cups used by Roman soldiers and other Romans for dice games. And those dice I saw this past fall at a museum in Dalmatia in Croatia from a Roman ruins dig. And there they were, dice just like you and I would play with. And they would rock them around inside these dice cups. So what I harvested were green capsules like that, which was easy to see the seeds and count them. And what I found is that unvisited flowers and self flowers, but especially unvisited flowers, set no capsule. Unlike death camas, in which selfing was not very effective either, in the case of checker lily, selfing, outcrossing, open visitation, supplementation, they all set lots of capsules, the vast majority of flowers, and they're full of seeds. Those ones that there were no visits, where there was a capsule set, it was frequently empty or only had a few undersized seeds. So it needs a pollinator, but not fussy about where it comes from, as long as it gets pollen of its species. It's a lot of seeds, neighborhood of an average of 80, as many as 140 packed into that capsule. And for all that seed production, it does not seem that this plant has much of a seed bank. And that's because in the autumn, after a little bit of chill weather, the embryo starts to elongate that first year. And after going through a winter of cold, wet stratification, it germinates. So the seed is committed. It's sort of like parsnips in that regard. And so I don't think there's much of a seed bank. So I planted a lot of what I got from all of my pollination experiments, both in my yard, some, as well as about 15 different outplanting sites here in Utah, as well as some colleagues did this in Idaho. And I sent some to Lawrence in England, where he's well on his way to having a common garden of all the fritillaria in the world. First year, a lot of my seeds came up. They look like little grass leaves. Really would never be able to recognize them in the field. The second year, I had some return and come up again. And I just saw today that I've got some third-year plants coming back. At the moment, it looks like 6 or 7 of them. So that's encouraging. It's also encouraging in terms of being able to put this plant back in the landscape, because I suspect it was more widely distributed before grazing, especially the intensive grazing in northern Utah prior to the Taylor Grazing Act back in the 1920s and 30s. So it begs the question that it does need a pollinator. It's not particular about where it flies from, doesn't need a dedicated pollinator. It just needs to go from flower to flower for a while. But who is pollinating? Well, in all my time on that hillside, bagging, unbagging, looking at plants, finding plants, I rarely saw any visitors. Sort of like orchids in that regard. And like orchids, one trick that people will do in order to concentrate pollinators a little bit, make a more attractive site, is to clip multiple flowers and put them in water picks in this case, and put them out as a bouquet. I also have some inexpensive cameras that allow me to do remote macro photography and time lapse. So I set up time lapse cameras on these with 6 to 8 hours of recording that I could then play back in about 4 minutes, looking to see who might be visiting, because I just wasn't seeing them. And also when they're visiting, because maybe I was looking at the wrong time. Turns out most of the floral visitation is in late morning. I have not seen anything visit in the afternoons. I haven't seen anything visiting before 9:30 in the morning on those days. Maybe it's just too chilly. And one of the first visitors that I got in these videos, and this is a still clip from the video, You might recognize that as a Polistes wasp, a paper wasp, the kind that makes the nests underneath your eaves. And it was probing for nectar. And like death camas, the nectar is not at the base of the stamens, but the nectaries are partway up on the tepal, both the sepals and the petals. A little shiny disc there that the wasp will be drinking from. And it's tall enough that as it's probing around inside the flower, madly drinking this nectar, it's rubbing against the anthers and presumably rubbing against the stigmas as well. You can see on the dorsum of that wasp, it's covered in pollen, and it's indistinguishable from death camas pollen from slide mounts that I made. So I suspect they are pollinating it. And you can see here, in fact, it looks like the plant can self-pollinate on occasions by how the styles continue to curl over and backward into the anthers if the pollen is still viable at this point. The flowers are fairly long-lasting. They last 6, 7 days at least. The puzzle with this is that and all the ones I've collected are European paper wasps that make these nests. They've effectively displaced the native paper wasp, at least in northern Utah. So it may have been that our native paper wasp in the past also visited these flowers and pollinated them. But clearly this is a newcomer to the role, but does it just fine. I also got occasional tachinid flies visiting the flowers. And you can see the fly on the left there, the dorsum of its thorax is as yellow as an anther with pollen. It's a carpet of pollen across its thorax. And you can see the dead one on the right. Those are all indistinguishable from fritillaria pollen, and it's pretty well coated. The problem with tachinids is the problem with nomata. Tachinids are impossible to identify, even the genus. They're horrid. In fact, they're hard to get the family. And the flies are like that. They're not very friendly at all compared to bees. So that's as far as I can get it as tachinid. So I had this odd array of things at this population near to town. I wanted to get out into a wilder place. And so up our canyon, I found a site that had been previously collected about 20 years ago. The plant had been collected. Turns out the GPS was incorrect, but when I went to their description of the site, I found a handful of plants, a couple dozen. When my wife and I walked in there the first time, hiked in, we found them in bud like this. So I knew I could come back in a week or so. We could come back and sit down, bring in lawn chairs. It's about a 1.5 mile hike in, bring nets, bring cameras, bring water, and sit down and watch. And you can maybe judge from my wife's expression there that after 3 hours of watching, it got pretty boring. We saw one visitor in 3 hours of watching 14 racemes. Nice bird song up there, pretty flowers. And the one visitor we did get, of all things, looks like a hornet, a bald-faced hornet, and indeed it's in that genus, but it's a social parasite, like Cythrus, the formerly Cythrus is in bumblebees. It parasitizes nests of other Dolicha vespula. And you can see it's covered in pollen, just like those Polistes were. And lo and behold, this is also a European import. And this is probably 15 miles from town or any habitation. So I continue to be baffled by what the native pollinators are for this plant, although it may be that it's going to be wasps like these, just members of the same genera. I've also gotten one yellow jacket. This was a native one. This is Vespula vulgaris, the common one, showing a friend one of these nearer population while we were out walking his dog. And the last flower we looked at as we were leaving the meadow, he saw the wasp, got a picture. I ran over with my net and successfully caught it. And there again, dorsum of the thorax, all this pollen. I've gotten exactly one bee, an andrena, and it has stumped both Terry Griswold and Skylar here. They can't identify it, although it's a female. If I got a male, I bet they could. But Skylar Burrows and Terry are unable to identify this specimen. So if you come across fritillaria, besides collecting these other things, watch for a bee. It would be really nice to know. And there's a precedent for that. That Fritillaria meleagris that I mentioned, that's common in parts of Europe I photographed it here in Sweden 30 years before I ever laid eyes on it in North America. I wasn't interested in it then, other than it's a famous plant in Sweden. But it's visited primarily by queen bumblebees in Sweden and some very nice research in Poland. So bees are within the realms of possibilities. There's no description of the odor of these flowers. My impression of Fritillaria atropurpurea at the time that wasps are visiting is a very faint mushroom smell. So not repellent. There's one in California called Stinking Bells that lives up to its name apparently and is horridly malodorous. But from what I've encountered with atropurpurea so far, it's just a faint mushroomy smell, and I might be wrong. So again, there's that website that you'll want to visit to look at fritillarias and get a little familiarity with them, because if you come across populations, I'm still working on the manuscript. I hope to have it done by this fall after I finish two others. But it'd be one that any additional insights about who's visiting the flowers, what pollen loads might be on them, how are they handling the flowers, all of that would be really welcome. Or the other fritillarias in North America, for which there's very little known. It's just a short clip showing what a male osmia does after it's drunk from a few flowers. It is an irritated bee, as you can see, and it seems to be that the tongue is irritated, probably its taste receptors, by the zygacine toxin that's in the nectar. And that is probably why there aren't piles of dead bees underneath death camas. Because any bee that forages at it other than Andrena astragale is irritated after the first couple drinks and then stops to groom, doesn't get poisoned either, and quickly learns that this is no plant for me to forage at. And that's probably also why I've never seen anything except Andrena astragale at the flowers. Actually, I take that back. I have seen one bumblebee queen one time, briefly. And then those flies, who are a bit of a puzzle at the moment. So that's what I have, and I'm glad to answer questions.
Jim, that was fascinating. I found myself getting more and more interested and awake as you were talking because it's such an interesting talk. We start out with Lisa, who asked, why can't we get Adrena astragalus renamed? Uh, Ornithologists are doing it. Uh, why has this bee remained the same name?
Well, it would take the effort of a systematist. You can appeal it to, there's an international zoological board to whom you could appeal for the name change. On the other hand, no one bothered to do that in changing the name of Peponapus first to Eucera and then to Xenoglossa either, even though it would be convenient since Peponapus means squash bee. But it's probably not worth the bother. There's a
Gotcha.
number of other plants that are like this, a number of bees, a handful of bees that are like this that were given a misleading name.
Understood. And we have a lot of questions, so we will be going kind of step by step, whoever came first, just so everybody understands. Uh, so the next one comes from Erica, who asks, "Are there any records or specimens of this bee in museums in the U.S.?" I'm not sure which bee you were referring to, Erica. So if you can clear that up for us, that'd be helpful. Erica, are you there?
I'm guessing she means the Andrena astragale, and yes, there are.
Okay.
It's also among Andrena, you notice those, which someone else, I think Kate noticed, the hair bands on the abdomen. That's not typical for Andrena, um, except for Andrena. And so
Hmm.
it's fairly recognizable amongst Andrena in general.
Let's see. Where are we at next? Lisa asked another question. What kinds of remote macro camera are you using?
They are, the, the brand is Mobius, Mobius, like a Mobius strip.
Hmm.
And they're they're a little tricky. They're a little bit bigger than a pocket matchbox.
Wow.
Um, so there's no screen. You have to memorize the light color. There's three little light diodes that you have to memorize, or LEDs, what they mean for starting, stopping, and which mode you want them in. And you have to set them up with your computer, set fixed focus points. And then you just measure the distance of the flower that you want to set up on. Mine are set at 2, 4, 8, and infinity for
Wow.
inches. But they're only $120 a piece. And the ones I've got have been working now for 8 years, I think. And so I can have multiple
Yeah.
ones. I can set them up one time lapse and one regular time. I can use the time lapse to find out and have them coordinated for time. So I can go to the time lapse first and find that, oh, at 10:42, there were visitors. I'll look at the other camera now for the good images on it. So they're Mobius. So they're fun.
Yeah. They seem useful and pretty economical, which is wonderful. Paige asked, nocturnal pollinators a possibility for the fritillaria?
Absolutely. Okay. And for that matter, as long as we're talking about for the death camas. I mean, it's a
Interesting.
white flower, right? But
Yeah.
I have not collected it either one at night. Um, Obviously, I can't camp on that hillside.
Yeah.
The place up at Limber in the mountains, I could. So that would be a possibility. The other places, there's a ranch in which people were a little frosty about me working there after I had led tours for them for a gathering. And the final place where I've encountered it is, well, two places. One is at which I might be able to camp there actually, over at City of Rocks National Reserve.
Mm.
It sounds like I can get a permit there fairly easy, and we found a population last year. So that would be a possibility. Absolutely.
To have a follow-up question to that, I was wondering, is that when I've been in Eastern Washington, I grew up in Eastern Washington, the death camas seem far less populous than normal camas. Is it
Yes.
is there a relationship to the amount of pollinators who are actively, interacting with death camas as to
Um,
the amount of death camas there are?
don't know.
Okay.
Um, for sure, I've never seen death camas in the abundance that camas meadow I showed you the picture of. That camas meadow was probably 5 acres
Yeah.
of solid bloom. What baffles me, and this hasn't been published yet, is that true camas, I was thinking that, well, it's a bulb, it flowers every year in the sites that I know, produces vast quantities of pollen and nectar. I would think that there's specialist bees on it, that there would be camas bees.
Sure.
And everything I've collected so far are generalists. Even the Andrena are generalists.
Huh.
So that kind of baffles me, but for sure it warrants more collecting.
Shannon asks, re fritillaria pollinators, uh, do you have any thoughts on phenological mismatch versus native pollinator decline, extirpation, extinction as hypotheses?
I don't, the mismatch argument in general, I think is overblown in that at least ground nesting bees in particular and plants are often using much the same cues for flowering and emergence. It's not perfect,